Now that we have discussed the main parts of any biosensor as well as the metrics associated with biosensors, we're going to start focusing on the types of biosensors according to the biorecognition molecules that are utilized to be able to select for the target analyte of interest. To begin with, there's some further reading that I would recommend. I have posted both of these in the track site actually, but you can also access this through the library if you're interested in some of the additional chapters that have to do with chemical sensors and biosensors. As a disclaimer, some of the notes that are included in this presentation were actually prepared by Dr. Weigand, and so I have modified them and added some additional information. All right, so to begin with, whenever we talk about an affinity-based biosensor, we're talking about the use of a biorecognition molecule that has the ability to bind to a target analyte through these non-covalent interactions. Specifically interactions that enable the formation of a molecular affinity complex. What a molecular affinity complex is basically a complex between the target analyte and the biorecognition molecule. So you can think about it like a lock and key kind of fit, where the biorecognition molecule would be the lock that has the right kind of cavity or the right kind of environment to enable the key to come in and click in, basically. These two molecules, biomolecules, would have the right kind of interactions. That could be electrostatic interactions, hydrogen bonding, hydrophobic interactions in the right locations to enable that kind of fit in between the two. Now, the strength of the interaction between the target and the biorecognition molecule is determined by the type and the number of such interactions. And again, those interactions are non-covalent, again, meaning hydrogen bonding, electrostatic attractions, etc. And because of that, they are also reversible. This is something that is important to remember whenever we have one of these affinity-based biorecognition molecules attached to the target. It doesn't mean that they cannot let go. We actually can change the conditions in which the interaction is taking place and might make that bonding basically break apart. So that's something to keep in mind. And that's also important because it would enable us to kind of reuse a biosensor, for example, if we're able to take advantage of this. All right. In terms of the specificity, that specificity is imparted by the steric and structural complementarity between the biorecognition molecule and an analyte, as I mentioned, kind of like a lock and key. Not every key is going to fit in every lock, right? It has to have the right shape, which is where sterics come into place, and the right structural complementarity, meaning that we have to have for example, complementary charges. Let's say that our analyte has a lot of positive charges, then your biorecognition molecule would have a lot of the opposite charge, right? If the target analyte has some hydrophobic components, then the biorecognition molecule would also have hydrophobic components for them to be able to interact, etc. Now, the biorecognition molecules that are used for affinity-based biosensors can be either of natural or synthetic origin. Most of the ones that we're going to discuss are natural, but I'll also give you just briefly a few examples of synthetic biomolecules. All right, so what are those biorecognition molecules that we use in affinity biosensors? The first are going to be these receptors. So receptors are proteins that are expressed typically on the surface of or the cell membrane of various cells. The role of these receptors is typically to convey information from the environment in which a cell is found in to the inside of the cell. So again, if this right here is representing my cell membrane, this receptor or this particular receptor spans the whole cell membrane. It has an extracellular component and an intracellular component. And typically what happens is that this receptor has specific affinity for some kind of molecule. Let's say this could be a receptor for the vascular endothelial growth factor. And so when the target molecule, which is the vascular endothelial growth factor, comes and binds here, then it would actually typically induce some changes on the intracellular portion of the protein that would then trigger a signaling cascade to induce some kind of change in the cell physiology. Here in a biosensor, really the only thing that we care about is the fact that this would be able to bind a particular biomolecule that it is designed to be able to interact with. The next type is going to be an antibody. Antibodies are also proteins, just like a receptor is. These antibodies are developed by our immune system, and I'm going to be focusing on antibodies on a number of slides. So for now, let's just leave it here. Let's just say that it has this kind of Y shape, and we'll discuss what all of the parts of it are in just a second. Nucleic acids can also be utilized as affinity-based biorecognition molecules. For one, they are able to bind their complementary strand in the way that is shown here, utilizing hydrogen bonding. But at the same time, they can be utilized to recognize other molecules through the use of aptamers. And we have a whole section on nucleic acid-based biosensors, so we will leave this one alone for now. And then on the right-hand side, I have two different examples of synthetic biorecognition molecules for an affinity biosensor. The first set, in this particular case, I'm actually showing cyclodextrin. So cyclodextrin is a polysaccharide that is synthetically made. And this basically acts like a host molecule that is able to interact with high affinity with certain molecules, typically hydrophobic molecules. Calixarane is another type of cyclic molecule like this as well. But yeah, so basically these act like hosts to be able to uptake some guest molecules, and you might hear about that as supramolecular chemistry, where these kind of large cyclic molecules will be synthesized to be able to interact specifically with certain other molecules. The last type that I wanted to mention are molecularly imprinted polymers, which we have discussed in the past. Remember that we call them MIPs. And this is basically a synthetic polymer network that is made specifically by using monomers that you might guess that might have some affinity for the target molecule. And so you prepared that cross-linked polymer network in the presence of the target analyte. And once you remove the target analyte, then you're left with a cavity that has the right shape, so the right sterics, as well as the right functional groups being presented to enable good interaction with the target. All right, so knowing all of this, now we're going to start focusing on antibodies on the next page. All right, so what is an antibody? An antibody is a natural component of the immune system. It is actually produced by B cells, which is a type of white blood cell. They belong to the protein class known as immunoglobulins, or Igs. They bind a unique epitope on the target molecules. So epitope is going to be the part of the molecule of interest that this is going to bind to. The molecule of interest is known as the antigen. The structure of antibodies is shown on the left-hand side. As I mentioned, they have this kind of Y shape. Among that Y shape, the top part is known as the variable region, which is what is now in white. And that variable region is the region that provides that antibody with the ability to recognize and bind to a particular target. So that variable region, as the name implies, is going to be different for any kind of antibody. That way it is able to then bind to a different target, right? So that part changes for all antibodies. Those antigen binding sites are also known as paratopes. And again, those paratopes would have the ability to bind to a particular epitope on the target antigen. Now from there, the remaining part, which is right now in gray in both sections, is known as the constant region. That constant region is actually conserved in between the various types of antibodies. So that's something that does not provide the antibody with a specificity. It just kind of provides it with the right structure to be an antibody. Now, the two parts of these antibodies are actually bound through these little dashed lines. Those dashed lines actually represent disulfide bonds that maintain the two sections together, and those can be dissolved by utilizing some chemistry, which we're not going to go into right now. Now, in addition to all of this, there are some names to the structure. So the part that I am highlighting right now, these BH, CH, CH, and CH, this is going to be what we know as the heavy chain. So that heavy chain contains both a constant region as well as a variable region. And then in addition to that, on the right-hand side, we have the light chain. So that light chain obviously is going to include both a variable region as well as a constant region. And those two parts get repeated on both sides. Now, the last thing that I wanted to mention in terms of the structure of the antibody is that this bottom part is known as the Fc region, and that region is the part that is recognized by the immune system. So basically, when this antibody is in your blood and the target, let's say, disease molecule that this is designed for, if it goes and binds to the disease molecule, then this part can be recognized by the immune system, specifically the B cells, and then the B cells can initiate the immune response toward that disease molecule, right, toward that microorganism or virus or bacteria or whatever it is. So again, this top part is what has the ability to bind to the target, and this bottom part is what would tell your immune system that that molecule or that pathogen has been identified. Now, in a biosensor, we would be utilizing this to be able to recognize specific molecules of interest. They might be a parasite, or they might be just another molecule of interest for some kind of disease or some kind of physiological response that we're interested in measuring. So of course, we're going to be needing this part to be able to provide our sensor with some specificity to the target, and then the other parts are not totally necessary. So you'll see that in some papers they actually utilize antibody fragments, where they might get rid of some of the parts of the antibody, or they might even just utilize kind of just this top part right here, depending on what it's needed. But really what we need is that variable region. And of course, utilizing this large protein, we have a lot of functional groups all along the chain that enable us to do chemistry to be able to immobilize that antibody onto a surface. One last thing that I want to mention about antibodies is that their molecular weight, at least for this one, is about 160,000 Daltons, and so they are very large molecules. They can be of up to like 10 nanometers in size. So that's just something to keep in mind. All right, I think that's all I wanted to mention there. Now, here I just wanted to show an example. Let's say that this is a bacteria. So this is the pathogen that we're trying to sense, we're trying to quantify. And you can see that the bacteria has a number of let's call them proteins on their surface. It could be proteins, it could be saccharides, it could be whatever biomolecule it has on its surface. So the antibodies that we're going to be utilizing in our biosensor are going to have some affinity towards some of those molecules on that bacterial wall, right? In this particular case, our red antibody can only bind to the triangular or cone-shaped proteins, right? While even though there's a number of other proteins, but this particular antibody can only bind to this. This other antibody apparently binds to something round, right? So each type of antibody would be kind of designed to bind a specific epitope on the pathogen that we are interested in. Because of the variability in the paratopes, you would bind to different epitopes on that target. And on the right-hand side, I'm showing just kind of another graphical example where this particular antibody has the right shape to only bind this particular antigen, even though the target might have a number of other antigens. But if you don't have the right shape and the right complementarity at the molecular level, then they cannot bind, right? That's what provides those antibodies with specificity. Their chemical composition and their shape that enables them to just be able to bind to one specific target. All right, there's actually a number of different antibody types. The one that I showed you is this kind of one, this one, which is typically the one that we most commonly utilize in biosensors. Particularly, IgGs are the ones that you're going to be seeing the most. So for the most part, IgG provides the majority of the antibody-based immunity against invading pathogens in our bodies. And because of that, it also has found the most use in biosensors. Again, this is kind of the monomeric kind of antibody. There are other types that have dimers, so those are going to be known as IgAs. And there's even a pentamer type of structure that has kind of 5 antibodies that are bound together into a large macromolecule. Now, one thing that does improve as you go from the monomeric type to the dimeric type to the pentameric type is that you actually have more what's known as avidity as you go down, because one of these antibody molecules would now be able to bind several of the target epitopes on the molecule of interest or on the pathogen of interest. And so you're going to be increasing the avidity or the ability of that antibody to bind. At the same time, we also have to consider the fact that as you go down, you're increasing in size, and so that might provide you with some steric problems for your biosensor or solubility problems or something like that. And so by far, IgGs, I would say, are the antibodies that are utilized the most. All right, so whenever you are designing your antibody and you go to a company website to buy an antibody, you're going to find that for any particular target, you're going to be able to buy both what are known as monoclonal as well as polyclonal antibodies. A monoclonal antibody is a particular antibody that binds one particular epitope of a pathogen or target microorganism or cell. Now, because it's only a single antibody type, it is going to have a single Ka or affinity constant for binding, and it binds a single epitope. The main thing that I want you to remember is that it binds a single epitope. A polyclonal antibody is actually going to be a mixture of different antibodies, and because of that, it is going to be able to bind different epitopes of the same pathogen. So let's say that I'm trying to develop a biosensor for the COVID-19 virus. I could buy, well, maybe, I don't know if they sell it yet, but in the future, I'm sure that we could buy a monoclonal antibody that would be able to bind just a single epitope, a single section of that virus. Or we could buy a mixture of antibodies that have been obtained from a mouse, for example, that all have the ability to bind to that virus, but on different parts of that virus. And if we want to do this kind of graphically, let's say that this is my virus, and a monoclonal antibody, let's say this guy, would only be able to bind right here. So for any kind of virus, it could only bind in this one section that has a particular protein or a particular saccharide or something like that. While if I go and I buy a polyclonal antibody mixture, then what I'm going to get might be one of this, but it also might be one that binds here and one that binds here, different type. Oops, it's not letting me. Here. All right, imagine an antibody there. And so even though all of those antibodies have the ability to bind to the same target, the same pathogen, we just need to recognize that a polyclonal one is going to bind to different parts. And so as you are designing your biosensor, you have to think about what makes better sense for my system. If you are doing a sandwich immunoassay, where you need to bind from 2 different locations, then you might be better off buying 2 monoclonal antibodies that you know are not going to compete for the same epitope. On the other hand, if what you're trying to do is just to bind as many antibodies to that antigen as you can, then a polyclonal antibody might be better because you know that you're going to be able to bind different epitopes and kind of saturate the pathogen. So you do want to keep that in mind. All right. So as I mentioned, polyclonal antibodies are a mixture of different antibodies. The way that they are obtained is actually by taking a small molecule captain, which is basically the target that you are interested in developing an antibody for. Let's say the COVID-19 virus or a portion of that virus, and you would typically inject it into an animal. The immune system of that animal is then going to start making antibodies toward that pathogen. And so after a certain time of infection, after infection in this case, you're going to be collecting blood from that animal and testing to see if antibodies are found. If antibodies are found, you would, or to detect whether antibodies are found, typically we're going to utilize an ELISA assay. In an ELISA assay, what happens is that you take a plate, typically like a 96-well plate or something like that, and you're going to immobilize the target of interest on that plate. Let's say our virus. So I'm going to put my virus, I'm going to immobilize it either by adsorption or some other method on the surface. I am then going to take the blood from this animal and put it in contact with this. And if there are antibodies that are specific for that target, then those antibodies are going to come and bind to my virus. From there, I then wash off anything that didn't bind. So all of my blood, all of the other antibodies that rabbit might have had are washed away. And so I'm left with just the antibodies that are specific to my viral target. And then you utilize a secondary antibody that has a label to be able to determine the presence of that primary antibody. And I should have used a different color, but whoops, too late. Maybe I can change that. Here we go, better. Yes, so then you utilize a secondary antibody that is labeled, let's say, with a fluorescent molecule, or typically for an ELISA, you utilize an enzyme to be able to do either a luminescence reaction or a just a colorimetric reaction that you can then read utilizing an absorption spectrometer or a luminescence or fluorescence spectrometer. And so if you do have antibodies against the target, then what you're going to get is a signal coming from the absorbance, in this case absorbance at 405 nanometers. On the other hand, if the molecule or the virus that you injected is not sufficiently immunogenic, then what you're going to get is a flat line on your ELISA. Basically, no binding occurs, and therefore none of our secondary antibody with the label binds, and therefore we get no signal. If this is the case, then what's going to happen is that we need to make this more antigenic. And to do that, what's typically done is that would be injected together with an immunogenic carrier. And there's different types, and I'm not going to go over that right now. But basically what we're doing is trying to present that target in a much more immunogenic way to the animal, so that once we inject it, now we're going to have more assurance that that animal will actually develop antibodies against it. And then once we collect the blood, we will be able to find some of those antibodies via the ELISA assay. Once we identify that we have antibodies, then in this particular case, we would start collecting those antibodies every so often from that same animal and be able to sell that. So ideally, you would want to continue selling that to be able to be utilized in a biosensor. And so one of the problems with polyclonal antibodies is that the mixture of antibodies that you get might not be the same from batch to batch, right? You're going to have changes depending on the antibodies that are being developed by that animal, and you don't really have control over the immune system of that animal. You just know that you're collecting a set of antibodies that are being produced by the animal and that have the ability to find that particular target, but you don't know what those antibodies are or what part of the biomolecule that or the virus in this case, it binds. You have no control over what the epitope is. but they're still antibodies and they still have affinity for your target. So they still have significant use in biosensors. Now, if you want a particular antibody that binds to a particular epitope, meaning a monoclonal antibody, then what you would do would actually be to go through this other process, where instead of collecting the antibodies from the blood of the animal, what you would do would be to still utilize an animal to inject them with the pathogen of interest or the target molecule of interest. You would then isolate actually the immune cells, let's say the B cells, which are the ones that are responsible for forming the antibodies. And once you collect those B cells from the animal, then you would fuse some of those cells with tumor cells, specifically multiple myeloma cells. And when you fuse those two, what you form is what's known as a hybridoma cell line. That hybridoma cell line has unlimited replicative potential because of the fact that now it is kind of like a cancerous cell. And it has the ability to produce the particular antibody that one particular B cell had the ability to produce. And so what you end up making is basically a machine, a biological machine that can produce that particular antibody over and over and over. And of course, being kind of like a cancerous cell, you can grow it, you can allow it to replicate, and you're going to have an unlimited supply of those antibodies. Of course, from here, we then have to collect the antibodies, but that is completely possible. So this is how you collect a particular monoclonal antibody, and that hybridoma cell line, again, can produce that same antibody over and over again. And so for that particular antibody, you would actually be able to find what is the particular epitope that it binds to, utilizing some biochemistry techniques. So we're going to be trying to detect molecules that are going to be present in a complex fluid, for example, a physiological fluid that might contain an array of other proteins, polysaccharides, cells, lipids, you name it. Because of that, we can still have non-specific interaction. Remember that our biorecognition molecule is the one that enables us to have some affinity. But if you have a bunch of other things that might absorb to the surface where your antibodies or your receptors are immobilized, you might still get a signal that is not specific to the target analyte. So because of that, we need to do something to minimize these non-specific interactions. And one of the things that is typically done is to utilize these blocking agents. So a blocking agent is going to be a substance that is going to try to prevent non-specific binding of other biomolecules that might be competing for them to basically not interact with the surface where the biorecognition molecule is found. Some of the agents that are utilized as blocking agents are going to be proteins. So one of the common things that you'll hear in or you'll see in papers is the use of bovine serum albumin or BSA. And so this might be something that you incubate your biosensor with before addition of your physiological fluid. Sometimes in addition, the physiological fluid might be further diluted in a BSA solution to be able to prevent non-specific absorption. Another protein that is utilized is casein, which is a protein found in milk, or you might actually utilize non-fat milk. So in a lot of biochemistry labs, they will actually have powder milk to be able to utilize for this purpose. Gelatin is another example that is used. In addition to that, we can utilize surfactants. Surfactants are basically detergent molecules that have both hydrophilic and hydrophobic components. And two of the more common ones are Tritonex and Twin20, which are both shown on the right-hand side. So Tritonex on the top, Twin20 on the bottom. And if you look at the chemical structure, hopefully you can recognize things that we have already learned in the class. So for example, this portion is polyethylene glycol, which is going to be water soluble or hydrophilic. And then the portion on the left-hand side that has carbons and an aromatic group is going to be the hydrophobic part of that molecule. Similarly, on the bottom, we have twin 20, which has several polyethylene glycol sections. But in addition to this whole side, like basically all of these sections that have polyethylene glycol are going to be hydrophilic. But on the right-hand side, you see that you have a carbon tail that is going to be hydrophobic. And so the role of these surfactants is going to be to interact with the surface in such a way that typically this hydrophobic part would attach to the surface and the hydrophilic part would then be presented toward the physiological fluid so as to prevent absorption of non-specific of molecules that we are not interested in detecting. And finally, another thing that you will see being done is to actually modify the surface of your biosensor with polyethylene glycol to provide a hydrophilic coating. So again, what we're trying to do is to kind of prevent non-specific absorption of molecules, proteins, or cells that are in our complex physiological fluid from attaching to that surface. And so to do that, what we want to do is to present something that is going to block all the sites All right, and so once more, the role of these blocking agents is going to be to prevent non-specific interaction of biological molecules with our surface where the detection is taking place. So for example, this is going to be my surface, and that surface is functionalized with antibodies that are going to selectively bind and enable us to recognize particular analytes, but what we want to do with either this kind of these kinds of proteins or surfactants or PEG is to be able to basically block the absorption of other molecules to the surface. So for example, the polyethylene glycol would be immobilized on the surface all around the antibodies to prevent other things from sticking that we don't want to detect, something like this. That's a terrible figure, I apologize, but I hope that you get the idea. So basically our surface now will still have the ability to recognize and bind to our target, but no other things could absorb easily to the surface. All right. So now we mentioned that the biorecognition molecules that are utilized for these affinity biosensors bind the targets through non-covalent interactions, and because of that, the binding is reversible. So that poses the opportunity to regenerate the biosensor, to be able to reutilize it after a detection event has taken place. And so to do that, we need to mediate the disruption of the binding between the biorecognition molecules, such as the antibody, from the target. To do that, what we need to do is to add one of these things or do one of these things. So we can change the pH, either make it acidic or make it basic. By changing the pH, you're going to be changing the ionization state of functional groups that are ionizable, such as carboxylic acids and amine groups, and that's basically going to end up disrupting some of the hydrogen bonding and some of the electrostatic interactions that you initially had between the biorecognition molecule and the target. Similarly, you can increase the ionic strength. Doing so, you're going to provide a number of ions or increase the concentration of ions that are available to compete with any kind of ionic interactions that you initially had in between your target and your biorecognition molecule. The third thing that you can utilize is to add guanidinium chloride, which is a molecule that is able to denature proteins. And so by denaturing protein, you are basically enabling some of the protein to unfold, that protein being your antibody or your receptor. And so if the protein unfolds, then the binding sites might not longer be available and your target would be enabled to leave. And so something that we have to think about these three is the fact that we are changing the conditions in such a way that you might actually end up completely denaturing your protein. So you want to be careful with this tree, because if you completely denature your protein, then that protein is no longer going to be able to bind your target, right? So you have to kind of play a balance between the strength of the treatment to regenerate the biosensor versus maintaining the activity of the antibody. And so that, again, you have to be kind of careful and be able to optimize the protocols that you utilize. Now, another thing that you can do is to actually add excess antigen to displace the initially bound antigen. And I'm trying to show an example of that here at the bottom. So for example, let's say that we have antibody, and this is a bad picture in the way that I drew it, but let's say that we have a surface, and that surface has a biorecognition molecule, which in this actually is our antigen. So I'm showing it as an ELISA, and I didn't think about this before, but we have a surface. On that surface, we have immobilized the target analyte. We then add antibodies, which in this case, we're trying to detect antibodies, I guess, and so binding occurs. But then we can add some more of that antigen or analyte, and because of the competition, that antibody is now going to bind to the more abundant antigen on the solution, as opposed to staying bound to the surface. You can use the same kind of idea. Let's say that we had a particular surface that was modified with antibodies, and let's say that those antibodies have now bound our antigen or our target molecule here and here. And then if I want to regenerate the sensor, one thing that I can do is to add more of those antigens. And so when I add the antigen, there's going to be competition for that binding site on the antibody, and you're going to end up displacing some of that antigen as well. So that's one way to be able to remove the antigen from here. Now, another thing that I could do is actually to add more of the antibodies. And if I add more of the antibodies, I can similarly displace my molecule here, because what would end up happening is that my initial surface, which has the antibodies, is going to stay there, is going to stay put. But now that other antibody that I added here is going to come and bind, sorry, it's not allowing me to draw very nicely. It's going to come and bind to the antigen and disrupt the initial binding. And so this is supposed to be showing an antibody binding to that triangle. I apologize, but I cannot draw it very nicely right now. But basically, by competing, we have removed the antigen from our biosensor. And so that's one way to regenerate, by competition protocols, basically. All right, now we're going to be talking about the transduction methods. And so there's going to be label-free as well as label-based transduction methods. We're going to first start with the label-free methods, in which case, basically, what we're doing is directly measuring the binding event between the analyte and the biorecognition molecule by utilizing either mass, electroactivity, or refractive index changes. So I apologize. In the case of mass, we can utilize quartz crystal microbalances or microcantilevers, which we mentioned before. These techniques are basically going to be looking at changes in the frequency of resonation or resonating frequency of these these surfaces as a function of the mass that is deposited upon them. The more binding that occurs, the lower the frequency of resonation, if that's a word, sorry. And so you would basically be able to detect the binding to that surface. In terms of electroactivity, certain analytes might actually be electroactive, and or the biorecognition molecule might undergo a change in structure upon binding, which might change the electronic kind of environment around your electrode. And so that can be detected as a change in the electroactivity, whether that's a change in the potential, a change in the current, etc. And finally, in terms of refractive index, we're going to be looking at changes in the refractive index of optical biosensors or surface plasmon biosensors. And I'm showing two of those on the right-hand side. So right here we have an optical fiber system in which light is going to be traveling through the optical fiber. We can actually come and modify the surface of that optical fiber to incorporate biorecognition molecules. And as more and more material deposits onto that surface due to specific binding, you can actually change the refractive index, and that's going to have changes on the light that goes to the other side, which you can then detect utilizing a photodetector. On the bottom, we have a picture of a surface plasmon resonance setup, which we had mentioned before, but basically the idea is that we're going to have a channel through which we can flow our sample. Then we're going to have a gold surface that is on top of an optically transparent material, and that gold surface can be modified with antibodies or receptors or aptamers or whatever biorecognition molecule you want to utilize. And as binding of the target molecule occurs onto those antibodies or biorecognition molecules, what's going to happen is that there's going to be changes in the angle of the light reflected because of the changes in the surface plasmon resonance here. And that is going to be optically detected. And this technique can be utilized to detect not just the concentrations of the analyte, but also the kinetics of the binding process. So that's something that is kind of unique to this process. And actually, you can also do that with the mass-based technologies, maybe with all of them. I'm not sure. It kind of depends on the sensitivity and things like that. All right. So what are the benefits of these label-free transduction methods? And let me back up. In this case, we are directly measuring the binding in that we don't need a secondary reagent to be able to do the detection. Just when the binding occurs, that binding already generates a signal. That's what label-free means. So what are the benefits here? Well, the benefit is that you have minimal steps and reagents that are required because the binding event is detected immediately. The disadvantages is that it is strongly influenced by non-specific interactions. So for example, we had talked about adsorption of other competing molecules to the surface. And so that adsorption is also going to be causing changes in the mass, is going to be causing changes in the refractive index, and in certain cases it might change the electroactivity of the surface, and so that can lead to incorrect readings. In addition, instrumentation may be more costly. For example, an SPR system or a quartz crystal microbalance, they might be fairly expensive instruments. And so that's something just to keep in mind, that it's not readily available for everybody. And then sensitivity may be lower. While these techniques are actually they have fairly good sensitivity. However, you don't have any way to amplify that each of the binding events. And so kind of your sensitivity is a little bit limited. With the other technologies that we're going to mention in a little bit, you have ways to amplify the signal, and so that can lead you to higher sensitivity. All right, so here are the label-based transduction methods. So we're going to have an additional reagent that is going to provide us the signal to let us know that binding occurred. One of the ways to do that is to utilize radioisotopes. So once your antigen has bound or analyte has bound to the biorecognition molecule, we could then radiolabel the biorecognition molecule, or we can radiolabel a secondary antibody to provide us with the signal that can then be read with a scintillation system. Of course, this is really not the best way. For once, you need to have the right equipment to be able to detect radio labels. You also have to work with potentially unstable radioisotopes. And so it's not ideal, but it is actually a very, very sensitive technology. So if you need very high sensitivity, this is something that would work, but it's just not readily available to most scientists. Optical labels, on the other hand, are readily used. And there are many types. And here I have just a couple of images of what's possible with each of these. So a colorimetric optical label would be one in which you use a chromophore or a chemical reaction that is going to cause the formation of a colorful molecule. So one example here is utilizing a sandwich immunoassay where we have a plate, like a 96-well plate, that has been functionalized with a primary antibody. That primary antibody is then exposed to the physiological fluid, which contains the pathogen of interest. That pathogen binds. We are then going to use another antibody that is labeled with an enzyme. That enzyme is not what gives the color, but that enzyme mediates the chemical reaction of a substrate, which in this case is TMB, or tetramethylbenzidine, which in the presence of the enzyme and in the presence of hydrogen peroxide would be converted into the cationic form of that substance, which is colorful. That reaction actually leads to the formation of a blue product, and that blue product is then reacted with sulfuric acid to give you this stable yellow solution that you can then detect using absorption spectroscopy. So colorimetric methods would be detected utilizing absorption spectroscopy. The second type is going to be a fluorescence-based label. And as the name implied, basically we're going to use a fluorescent molecule. And that example is shown right here. In this case, we have a different configuration, but it could be the same configuration, but with a fluorescent molecule here. But I'm showing just another configuration just to give you more options. So in this particular case, we are doing a direct ELISA assay where we have adsorbed our biological molecule of interest on the surface. We then add a primary antibody that is not labeled, but that has affinity toward that molecule. And then we use a secondary anti-IgG molecule or antibody. So this is a secondary antibody that has specificity for antibodies. And that one is actually, I'm sorry, I'm looking at the wrong one. For fluorescence is this one. I apologize. But same idea, we still have the molecule here, the primary antibody that is not labeled. Here's the secondary antibody, which has affinity for antibodies in general, and that's called a secondary antibody. It's an anti-IgG antibody, and that one has a fluorescent molecule. That fluorescent molecule needs to be excited with a excitation light source, and that molecule would absorb the light and it would emit part of that light in the form of fluorescence light of longer wavelength. And so yeah, that's your fluorescence signal. From there we go to luminescence, which is what I was actually showing. So in the luminescence transduction, what you have is similar to the first one, in that typically what you're going to label your antibodies with is an enzyme, and that enzyme could be something like luciferase, which would have the ability to take a substrate like luciferin and enable it to react, and in that reaction light is going to be one of the byproducts. So basically you end up creating light through a chemical reaction, and that light can be detected by a spectrophotometer. Fluorescence is read by a fluorescence spectrophotometer, like the one that you guys utilized in my lab. Luminescence can be read with the same spectrophotometer. And then from there, we're going to go to nanoparticle aggregation. So this is something that you will see in papers that utilize sometimes gold nanoparticles, for example. And the fact is that gold nanoparticles, whenever they are in their monomeric form, they're going to have a different SPR signal than if they are in their agglomerated form. And so if you can induce agglomeration as a function of target analyte, then you're able to detect changes in the absorption spectra of those singlets versus, or not singlets, but those individual nanoparticles versus the aggregate of nanoparticles, as is shown here. So in the single nanoparticle state, you're going to have absorbance or an SPR peak that is more blue shifted, versus the aggregates are going to be typically red shifted to show you a new peak here. And that can also be actually seen through, through by observation. You'll see changes in the color of the suspension of nanoparticles. Similarly, here on the top right, I'm showing aggregation of other types of particles, not gold necessarily, but basically aggregation of particles could also be looked at through scattering, so the scattering of light. If you have individual nanoparticles that are smaller than the wavelength of the light, then the suspension of the particles would look transparent. But if the nanoparticles aggregate into now aggregates that are larger than the wavelength of the light, then they're going to scatter the light and they're going to appear cloudy. So this could also be detected with an absorption spectrometer, but it could even be detected by eye, as you can see here, from a clear to a cloudy suspension. Now, I mentioned that these label-based transduction methods have the ability to be very sensitive because of amplification. And so the amplification could be that, one of the things is that you could label your antibody with more than one of those enzymes or those fluorescent molecules, right? You could have more of those fluorescent molecules, for example, and that would increase your sensitivity. Similarly, the binding is not necessarily a one-to-one as is shown here, depending on what your target is. If your target is large and it has various epitopes for which you have different antibodies, you could actually have several antibodies that are binding to the one pathogen, for example, and that would result in a signal amplification, of course, for any of these techniques. So that's just something to keep in mind. All right, so what are the advantages of label-based transduction methods? Well, the first thing is the high sensitivity. The second is the flexibility in terms of the label selection and instrumentation. For label selection, as I mentioned, you have fluorescent ones, you have enzymes, you have different substrates for those enzymes, and you're going to find hundreds of companies that sell these various agents that are already labeled, or even labeling kits for you to be able to do this yourself. So there's lots of choices. And in terms of instrumentation, most of these are going to be utilizing absorption or fluorescence spectrophotometers, which are typically readily available in most universities and research and development places. So it's something that is accessible. The disadvantages, first of all, the conjugation of the label to the biorecognition molecule can sometimes alter its ability to be able to bind to the antigen. For example, if I'm trying to label my antibody with a fluorescent molecule, I can label it with many of these fluorescent molecules by utilizing chemistry, for example, that would utilize some of the primary amines on that protein. But I don't have really the ability to just select this one amine group versus this other amine group. I'm going to be labeling wherever I have amine groups. And so sometimes that means that the fluorescent molecule might end up blocking or changing the binding site of that antibody. So that would basically now make it inactive or prevent it from actually binding to the target of interest. So that's something to keep in mind, that if you label in the wrong places or put too much of the label, then it might decrease the activity of that antibody or that biorecognition molecule to be able to bind to its target. Now, transport proteins, what I mean here is that if you put too much of those labels, you might change the solubility of the biorecognition molecule. And so if, for example, let's say that you were trying to do in vivo detection, utilizing some kind of nanoparticles that were labeled with an antibody. If that were the case, by putting too much fluorophore or something on that antibody, you might change the hydrophilicity and the ability of that particle to actually cross cell membranes or be able to circulate in the blood for a period of time. So you will be changing its transport properties. All right, in addition to that, we need to consider interference from endogenous molecules, meaning molecules that are already present in the physiological samples or in the patient. One of those things is autofluorescence. So there are certain proteins that are known to have fluorescence because of the presence of a certain amino acid that is fluorescent. But typically that autofluorescence is fairly low. So this is not a huge deal. However, if you do find that autofluorescence from the biological sample is an issue, one thing that you can do is to go to a luminescence-based assay. In a luminescence-based assay, you're not going to have any, I should say, because luminescence doesn't require an excitation light, it's just going to be detecting whatever light is emitted from the chemical reaction, so you don't have any background light. While in any kind of fluorescent detection method, you need an excitation light, and that light might excite not just your label, but might excite some of the proteins that were already in the sample, right? So you can always switch to a luminescence assay. Now, if you're using a colorimetric assay, which is the ones that produce a colorful molecule that you would detect by absorption spectroscopy, you might have interference from other biological chromophores, such as hemoglobin and oxyhemoglobin, which are going to absorb in the visible range, melanin, which absorbs in the visible and near infrared range, and proteins and DNA that absorb in the UV-vis range. So that's just something to keep in mind, and by utilizing the proper controls, you should be able to take care of it. And then finally, if you're utilizing enzymatic transduction, then the presence of endogenous peroxidases or endogenous enzymes that might actually react with the substrate that you utilize, they might create some background signal. And so typically, again, you can either change to a different kind of technique, or you can utilize proper controls to see what would zero concentration of my analyte actually look like. How much fluorescence do I get if I have zero of my analyte? How much luminescence would I get if I have zero of my analyte? And from there, be able to detect your sample. All right, last I'm going to go through a couple of assay formats. So first assay format is going to be a direct immunoassay. In a direct immunoassay, you're going to be immobilizing the antigen that you're trying to detect onto a surface, and you're going to be directly detecting that antigen by utilizing a labeled antibody. So it's kind of a one step, just one antibody. That antibody has a label immediately, right? And I can immobilize my antigen directly onto the surface as is shown here. Or here I have actually a sandwich immunoassay that is direct. So let me hold on for a second and tell you what a sandwich immunoassay is before I go to this one. So sandwich immunoassay is one where the antigens are going to be sandwiched in between two antibodies. One of the antibodies is going to act as the capture antibody that basically is going to now immobilize that antigen. And then the secondary or the second antibody is called the detecting antibody. And that detecting antibody basically is going to bring either directly is going to bring the label, or is going to enable us to bring another secondary antibody to be able to give us the signal. In the direct sandwich immunoassay, you're going to be, again, utilize your capture antibody and your detection antibody, but the detection antibody is directly labeled, just like in the direct immunoassay. So basically, the capture antibody provide us the way to immobilize that antigen, just like kind of like the way that we had here, except that here it was adsorption. Here you actually have an affinity interaction to immobilize the antigen. On the indirect method, you still have the first capture antibody to immobilize your antigen, and then you utilize two antibodies for the detection. The first one is unlabeled, but it has affinity to the antigen. And then the second antibody would be an anti-IgG, in that it is specific for binding antibodies, and that one brings a label. And those labels can all be either enzymes, they can be fluorescent molecules, they can be, what else did we talk about? I guess, yeah, you need enzymes to be able to do luminescence or fluorescence, or you could have a fluorescent molecule. All right, so the third format is a little bit harder to explain, and this is known as a competitive immunoassay. In this case, the analyte is labeled and is added to a sample at a fixed concentration to compete with the unlabeled analyte that comes from the physiological sample for the binding sites of the biorecognition molecule. So let me just kind of back up. You have a biological sample, let's say blood, and that blood has a particular analyte. That analyte in the blood is going to be the one that is grayed out here. So this one or this one. That's the one that we're trying to detect. Now to detect it, what we're going to do is that we're going to add a tracer analyte at a known concentration, and that tracer is going to be labeled either by fluorescence or with an enzyme or something like that. And so in the left-hand side, I have a competitive direct assay where I have a transducing surface, let's say an electrode. That electrode has an antibody immobilized, which acts as the biorecognition molecule. And then I add both my blood that is spiked with an analyte tracer. And so what's going to happen is that in the presence of the analyte, I'm going to have less of the tracer actually binding to the antibody than if I didn't have the analyte. If there was no analyte, then all of my analyte tracer would actually bind. And so it's going to give you kind of the opposite signal that you expected. So let me kind of plot here. I'm going to have concentration. Oops. Okay, let me write. Come on. Concentration versus signal. So my x-axis is concentration. My y-axis is whatever the signal intensity is. That could be fluorescence or luminescence or chromophore concentration or something like that. Or absorbance, chromophore absorbance, I should say. So if I have no analyte, I'm going to have the highest signal from my tracer because the tracer would be able to bind. There would be nothing competing with it. So zero concentration, high signal. And then as I add more of my analyte from the blood sample, that one is going to compete and is going to prevent my tracer from binding. So what you're going to see is that the concentration increases, the signal is going to decrease, if that makes sense. On the right-hand side, I have competitive but direct method. So in this particular case, the transducer is actually going to be bound with analyte. So this is the analyte that you purchase and that you know the concentration that you are binding to that surface kind of thing. Now, the analyte here is the one that comes from your blood sample. What we're going to do in this particular case is that actually label the antibody or label the biorecognition molecule, let's say with a fluorophore. And so imagine that you have none of these analyte from the blood. Let's say that the blood doesn't have the analyte. So what's going to happen is that antibody is going to be able to bind to the immobilized tracer molecule or immobilized fictitious analyte here. But if I have some of the analyte from the blood, which is not immobilized, what's going to happen is that some of that antibody is going to bind to that analyte and then it can be washed off. And so some of this is going to end up being washed off with the analyte. And so again, what we're going to have in terms of signal, as the concentration of the gray analyte, which comes from your biological sample, increases, you're going to have less and less of that primary labeled antibody actually binding to the surface, to that immobilized tracer analyte. I hope that this makes sense. And if it doesn't, give me a call, send me an email. All right. Last but not least, I have a couple of slides on common platforms that are utilized for biosensing. One of them is the ELISA assay that I already mentioned. In this particular case, however, I'm not showing the direct ELISA assay that I showed whenever I was talking about that viral detection number of slides ago. In this particular case, I'm using a sandwich ELISA assay. And so in a sandwich ELISA assay, what happens is that you first immobilize a capture antibody. You then add your physiological sample, let's say blood, and if that blood contains the target protein, then that protein would bind to the antibody. Then you're going to utilize another antibody, which is going to be the detection antibody, which is going to enable you to provide some signal. In this particular case, what we have is biotin, which is actually not giving a signal, but is enabling us to bind to something that will enable us to give signal, which I know it sounds complicated, but here is just providing us another molecule that can enable some binding to take place. And that's just kind of to make the assay a little bit cheaper. So instead of labeling this antibody directly, they label it with biotin. Biotin can then bind to streptavidin, and that streptavidin was now labeled with the enzyme. So this is my horseradish peroxidase. This is my enzyme. And yes, I could have done that by directly binding that enzyme to this antibody, but in this particular case, they chose not to. Anyways, now that my detection antibody has the enzyme, we can then add the substrate. And in this particular case, for ELISA assays, typically that substrate is TMB, tetramethylbenzidine, and that substrate reacts in the presence of horseradish peroxidase to form a blue reagent. As the reaction continues, since it is an enzyme is catalytic, so the reaction actually continues and continues and continues. You actually have to stop the reaction by adding sulfuric acid, and when you do that, the reaction stops, and this blue colorful substance actually changes to a yellow color. And so a yellow color here would indicate that you actually did have this target protein. If the target protein was not here, then whenever you add this detection antibody, there would be nothing for it to bind to, and so it would be washed off. There are a number of wash steps. Pretty much every time that you add something, you wash. From here to here, you add target protein, then you wash. Then you add the detection antibody, then you wash. Then you add the streptavidin with HRP, then you wash, and then finally you do the last part by adding the substrate. This is the assay that is utilized for the detection of HIV, for example, and it's also the assay that is likely being utilized nowadays for trying to find antibodies from the blood of patients of COVID-19 and many other diseases. All right, another common platform is going to be the lateral flow immunoassay, and most of you have probably heard about this immunoassay from pregnancy tests, but it is also utilized for a number of other detections of biomolecules. The way that this works is that you're going to have a kind of membrane-based system where we're going to have an adsorbent pad whose job is to try to weak solution from the left to the right. And we're going to have a membrane here. That membrane is initially labeled with antibodies here at the test line and another set of antibodies here in the control line. These two antibodies are different. They bind to different molecules. The control antibody actually is specific to bind to IgGs. That's why it says an anti-IgG. It's an antibody that is specific to other antibodies. The antibody immobilized here in the test line is an antibody that is specific to the target analyte, the molecule that you're trying to detect, which in this case is this red thingy. From that membrane, we then go to a conjugate pad. What that conjugate pad has before you begin is actually an antibody, which is going to be the label antibody, and that's basically, it's not immobilized. That basically, I should say that, that labeling antibody is going to be dried on that pad. So it's not bound, it's just kind of dried in place. And then finally here we have the sample pad. The sample pad is where you would put the physiological fluid. That could be urine, that could be blood. And that physiological fluid will likely have the analyte that you're trying to detect. And so once you put that physiological fluid here, what's going to happen is that it is going to start traveling through capillary action toward the right-hand side where you have that adsorbent pad that acts as a driving force, right? And so when the physiological fluid gets to the conjugate pad, it is going to now dissolve the labeling antibody, and this guy is going to bind to the antigen, to the red dot. And they're going to travel together. Once they get to the test line, now that antigen is going to be captured by the test antibody here. And you're going to end up with a signal here. And that signal is typically chromophore, a color that you can see with your eyes. And then more of the sample is going to continue through. And here you're going to have basically more of those conjugates that didn't bind to any of the targets. And so they would continue and they would bind here where we have that antibody that is specific to other antibodies. And so you would end up with 2 lines. And then the extra fluid would continue to the adsorbent path. And so a positive result would be one where you get 2 lines on that assay, indicating that yes, you have the red dot analyte that you were trying to detect, and yes, the whole assay worked enough for the fluid to continue through and get to the control path right here. If you don't get both, if you don't get signal here in the control line, then that means that the assay probably didn't work. Either the whole thing kind of stopped here and didn't continue flowing, or the antibody all stayed here in the conjugate path and something just went wrong. Now, if you don't get a line here, but you do get a line here, that means that the assay worked, but there was no analyte, nothing to capture, right? So everything basically just continued through. Now, typically the label that is utilized for lateral flow immunoassay is actually going to be gold nanoparticles or something that can provide visual cue without the need for a spectrophotometer. But of course, there are many other people that are developing systems that might utilize like a fluorescent molecule to provide you with a lot more sensitivity than you can with your eye. And that could be utilized in conjunction with something like a cell phone camera to be able to detect something with more sensitivity, I should say. All right, the last platform that I wanted to mention are microarrays. In a microarray, what we're going to end up doing is trying to detect the presence of a range of different proteins. So you can think about it like a multiplex biosensor that is going to detect many proteins or many antigens from your physiological fluid. And so what you do is that you take your physiological fluid, say blood or serum or tissue or urine, and you're going to extract the types of biomolecules that you're interested in. Those could be proteins, as shown right here, or they could be nucleic acids, depending on what you're trying to detect. And there's various protocols to be able to isolate proteins versus nucleic acids. You're then going to add fluorescent labels to those molecules. Now, in this particular case, it is showing like, actually, I should say that. So you don't have specificity here because let's say that they're all proteins. All proteins are going to have primary amines. All proteins are going to have carboxylic acids. Most proteins are going to have thiol groups. And so when you add your fluorophore, most likely you're going to end up labeling all of your proteins with the same fluorophore. So at this point, you don't have any specificity for one protein versus the other. But then separately, you're going to prepare your antibody immobilization surface. And so what that means is you're going to take a surface and you're going to immobilize specific antibodies in specific locations. That can be done utilizing a microarray printer that can go and deposit very small volumes of antibody solutions, different antibody solutions into different spots, as is shown here. So each spot would have a different type of antibody. And again, that can be done utilizing a kind of printing system or a small volume deposition system that can do it automatically. Or you could have a student just pipetting 2 microliters into different dots. Just kidding. All right. Sorry. It's late. So from there, what's going to happen is that now you're going to put the two things together. So you have your array that has antibodies. You need to know where you put each of the antibodies and what they are specific for. But then you put the two together. And so if your physiological fluid has that particular antigen, then that antigen would bind to the antibody. So in this particular case, my red antibody is specific to the red molecule, and you can see that the binding takes place, and the green one is specific to the green one, and so on and so forth. And so now you have specificity. Now you have one molecule binding to one location on that array. And after that, you can then do you can obtain an image utilizing fluorescence. And in that image, you're going to have basically a specific location, you're going to have more or less fluorescence intensity. The higher the fluorescence intensity, it means that you have more of that analyte present. And the lower it is, then that means you have less or none of that target molecule. And so by knowing what antibody you put in what location, you can then know what analyte is present and more or less in one concentration. And after that, you can then utilize other techniques to be able to validate your assay. For example, Western blots or ELISA assays or immunohistochemistry or even literature search, just to kind of double check the results that you obtain. But utilizing this, you have the ability to now identify a number of different proteins in a very complex fluid. And so this is a very powerful technique. All right, that's all that I'm gonna tell you guys about in terms of Affinity biosensors. Bye-bye.